So let me start by thinking about what biodiversity is. Okay, as Richard has thought, we both work with large scale patterns. Okay, and biodiversity actually is the largest pattern we study in biology, arguably. Okay, and I really like the title of this Kevin Gaston's book is A Biology of Numbers and Difference. If we look to the numbers, which is how we often think about biodiversity, we first get to this idea of this big bars of how many species do we know and how many species may be left to be known. Okay, you see huge gaps, not so much for vertebrates and chordates and plants, but we see huge gaps. We don't know a large part of those numbers. But biodiversity is more than that. Okay, biodiversity is all this variety of forms, functions, and differences between even individuals across all the scales of taxonomic, all of the tree of life. Sorry. Okay, in fact, what we study in biodiversity science is this whole tree of life and how these relationships and how these phylogenetic relationships are affected by and affect, within brackets, the distribution of the species. This is a map of the amphibian richness, the different functional traits and functions that the individuals and populations perform, and how those individuals, populations, and species interact in Earth. Okay, all of that is the subject of biodiversity science. Okay, deciding or trying to understand how much of this whole variation, not the number of species we don't know, as we saw in the bar, but how much of those variation of this variation we are yet to know. We have to go back to the philosophy of knowledge in science, okay, and to the concept of scientific ignorance. Scientific ignorance, as defined by Maxwell, and it's the quote is for this nice book of Joaquin, I recommend you to read, is the prelude to every real advance in science. In a way, this is what Socrates was saying when he said, the only thing I know is that I don't know. Okay? If we want to know something, if we want to reach to understand something, we first have to recognize that we need to know something, that we don't know something of it. If we believe that we know everything, we are not in the position of learning nor of studying. So scientific ignorance is this kind of approach where we make ourselves conscious that we ignore a lot of the natural world. Okay, and what Firestein says is to use this pond idea when you throw a stone to the middle of the pond, and it starts to create waves around. This is what we know. Imagine that this is the universe. Okay, this is what we know, and the successive front of this wave is what we now call cutting-edge science. Is the limits of our knowledge. Okay, those limits are expanding continuously as we do scientific research. In biodiversity, as we describe new things, describe new interactions, new traits, new functions, or measure them. But it creates the paradox that the more we know, the more we need to know, because the borders of knowledge are limited. Okay? What Firestein is arguing for is that actually the limits of this knowledge are not just simple waves. They look this way. There are many areas in the limits of knowledge, okay, where we have bits of knowledge getting into the unknown, okay, and bits that are less developed. And what he's saying is that we are able to identify these small areas. In the in-between, we have more opportunities of making fast discoveries with less effort, filling in the gaps. A good example of that is, for example, the Higgs boson. You know that particle that was described 20 or 30 years before being discovered. What Biggs and colleagues did, with Higgs and colleagues did, was to put together all the characteristics of the particles at the time, and decided and said that either we have a particle here with these particular characteristics, or we have to rethink all of our particle theory. Unfortunately for physics, they find it. They put together a great effort and they may find it. I'm saying that completely because it's better when you don't find what you expect and you're open to new discoveries. But that's one of the ways of advancing science, identifying of what we know and identifying these little gaps that we can fill in relatively easily compared to going here, which is a subject of sci-fi or really, I mean, we need a lot of effort to continue expanding in these ways. Okay. So going back to the science of biodiversity or biogeography, as I used to call it, so I'm a biogeographer. Okay, this is our dream data for all the analysis we do. This is imagine a magnificent and omniscient GIS. That is a paper from Caldwell, and John Coddington a while ago, where we have all the information on living species with the capacity to display any part of it at any moment of time or any moment of the year. This metaphor is quite useful, not because we are going to be able to reach it, but because it defines the whole poem, the whole universe we want to know. We want to know all the species, where are all the species, all the individuals, how they interact, etc. That this is impossible to know, but it is our main goal. Okay, that it is impossible, it doesn't mean that we don't have to try to reach for it. And for us, it provides a way of defining the universe we are working on. Okay. And we are actually working towards knowing all of this with all of these big databases. When we integrate all information and put in KGBs, but all the Open Trades Network, this what is it there? This network of gene bank of another genetic databases, map of life, and there are many, many examples out there of databases where we are sharing in most cases openly all of our knowledge. So anybody can retrieve it and use it. Okay, we are trying to approach that omniscient GIS. Okay, when we put together all that information, this is what Richard was talking about. We have we may have the impression that we know a lot, and it is true. We do know a lot. We know a lot of things, and we have recorded a lot of things, and we have mobilized a lot of information. Mobilizing is what we do from retrieving information from journals, from collections, etc., and putting it available online, okay, or available for everything. We have mobilized a lot of information, but that information is not complete. And many, many, many times we work like that information is complete. No, it is not. It can never be. Okay, but that my argument is it can never be, for our argument. And that is why we decided to get together when we had the opportunity of writing an annual reviews paper. And this was done at Richard's home at this paper. So, I mean, at least defined. Get together and put all of the main aspects of ecology together, identify shortfalls for each one of them. Some of them were described. We continued the unfortunate trend of naming them after all white males that are all dead. Okay, because that's how, and unfortunately, those are the biases we had at the time, and the historical biases in research. But forget about the personalities here. We are looking at 7 different aspects of biodiversity. The species, called Linnaean, so we have not described most species on Earth. And also we can extend that to the distinct species. Of course, we are not going to be able to describe all of them. It is virtually impossible. Okay? But being aware that we've got a gap puts us in the position of being cautious each time we need it. Geographic distribution, we don't know the distribution of most species. Populations, quite important data on population. And population trends is really scarce. That was described by Pedro Cardoso and colleagues. Evolution, we don't know about the tree of, we don't know the complete tree of life, and we have problems to identify the right models of evolution or to describe the evolution of all traits. The Raunkiaeran, its functional traits and ecological functions. We don't know all the functions of the individuals, and we don't know how the traits relate to those functions. Abiotic tolerances, we don't know that, and ecological interactions. Okay. The main idea of that is to identify the 7 main aspects of biodiversity research and put shortfall out of it. Recognize that we don't know. We are recognizing our ignorance as a first step to improving our knowledge. Okay. One thing you discover when you think in that way is that they are all interlinked, and we were not discovered anything particularly surprising. But if you put them together, one of the first things that came out is the Linnaean shortfall. Is the one that binds them all and ties them to darkness. Okay. If we don't know the species, we cannot describe the interactions, and we cannot describe the functions, and we cannot describe the phylogenies in a good way of the evolutionary relationships. Okay, so this is this taxonomy impediment may stop us from doing cutting-edge ecological and evolutionary research. So we need to invest in taxonomy. Okay, but if we invest too much in taxonomy, like here, we may have a good knowledge of how many species are there, of the numbers, but with little content, these different shades of orange indicate the levels of, I mean, from the core ecological perspective to the large scale macro, to just knowing how many species are there on Earth. Okay, so if we just invest in taxonomy, we are going to miss the functioning and evolutionary patterns. If we put a lot of money in ecological and evolutionary patterns, we are going to be, our ability to grow is going to be stopped by our lack of taxonomic knowledge. We need to find a balance between both. And it's important that we say this out loud in many contexts, because as you know, we are not giving taxonomic efforts the importance in terms of funding that other aspects of biodiversity research are receiving. Okay. Another important thing is that the gaps in our knowledge may make us see things in very different ways, even with the same, even with gaps of the same size. Okay, let me explain that. This is a phylogeny, a mock phylogeny of lizards. And imagine that we have only known 4 species. If there is a bias in the species that we have known, imagine in this case we will only know the brown small lizards and the blue small lizards. Okay. Note here that trait evolution through time is constant. If we look at this, okay, the 2 correlated base, it goes down a bit. But our perception of the relationship between body size and color throughout the phylogeny is completely wrong. Okay, there is no relationship, and here we believe that, of course, color and body size are correlated only because we have a bias. If our bias is that we miss clades because, for example, this another clade is in a different island that we have not surveyed on a different territory, okay, we may get a glimpse of this as well. But our patterns or our perceived patterns of trait evolution may be quite different. We can believe that we had a fast evolution. We are missing a lot of this part of the tree. Okay, and even if we have an evenly distributed sampling of the species, we may have slight deviations. This is a 4 slight deviations from the actual pattern. Okay, but that's the thing. The different shortfalls may be interacting in very different ways. So depending on the kind of a study that we are conducting, we have to look not only at the shortfalls that may affect us, but also to the interactions between them. And some of the examples I'm going to provide now, the things we are doing, go towards this direction. Okay, so what can we do with this thing? It's not that to say you know nothing, just know. Okay, it's not that we don't know nothing. We know a lot, but the awareness of the things we don't know should lead us to think and describe that problem. This is a paper by Justrop and I mean we did with Juliana, where we mapped the coverage of knowledge about seed plants in Africa. Okay, and this is the completeness. Okay, here B. This is the number of cells with progressively more complete inventories. The reds are the good ones. The white is no data, and this one is having very few data. As you see, there is a bias, a geographic bias there. Okay. South Africa is much better known in certain territories. But if you look to this is the year where the data was gathered. Our temporal coverage in South Africa, those areas is limited because it's all old data. Okay, so if we are working with changes in the landscape, we may not have data for the place where we think we have good data. Okay. This is another study we have done recently in the Atlantic forest, a group of butterflies, fruit feeding butterflies, and what we mapped there was the coverage along environmental gradients. But since we are working at the Atlantic forest, we are not only covering the climatic gradients in the Atlantic forest, okay, climatic gradient. We are also looking at the landscape characteristics. Okay, we are looking at the gradients in the size and shape of the remaining fragments because it's key to know how this group of butterfly species will respond to changes in land use. And what we actually identify is we've got a little coverage of intermediate and small fragments. Okay, so we have a good coverage, relatively good coverage, it's not here, but a relatively good coverage of climate instead of the rare climates, okay? And we can make models out of that, but we don't know the response of those species to landscape fragmentation because we don't have good data of intermediate to small fragments, okay? So this is tailoring our description of the coverage of knowledge to the particular questions we are trying to address with the data. In this case, the effect of land use impacts. And this is a paper that is already in press. Here we are looking at population data, okay? Looking for all the data that we've got in amphibians and reptiles, or data that we've got about paired sites with pristine or natural habitat and anthropized habitat, about the population trends, making a space by time substitution, okay, between the state, the population size or population dynamics of the communities in natural area, in natural fragments, and in anthropized, in fragments affected by human activities. Okay, and what you can see here is the distribution of the differences between the total reaches of a species and the total reaches of a species with abundant data. Okay, we have deficits of abundance data here, and we have excess. We have better quality of habitat data in these areas. Okay, we have an excess of the species compared to the rest of the world, of species with an excess of the species with data on the effects of land use change on populations. Okay, here you can see that these effects are contextual. to each group. Okay, so here the red residuals are more in Asia and Oceania than here. Okay, in the case of reptile. Okay, this is another example working with fish data. It's a group of catfish from the Neotropics, the thesis of the PhD of Thiago Freitas. And what he was interested in, what he was working with PhD was about the evolution of feeding habits on those catfish. Okay, so we look at the shortfalls that affected his question. First, about the phylogeny, because we are working with evolutionary patterns, and we look at the stability in the phylogeny, the similarity between phylogeny. We retrieve the phylogeny from all the matrices of distance between species, and we track how similar they were. You see that there are a few phylogeny that are relatively similar, and then we look at the coverage of the total valid species at each time, at the time the phylogeny was published, and the coverage of the species with phylogenetic information. And actually, we could conclude that this Calegari and colleagues phylogeny was quite good and was covering a lot, so we could use it. Okay, but then we look to another shortfall, the availability of data on feeding habits. Okay, so we look at the accumulation of species, the accumulation of species with feeding habits for which the feeding habits have been described. Okay, and what we see here is that, well, we are still accumulating. There is a gap of knowledge there. So when Thiago was interpreting results, he has to bear in mind that for particular groups, we identified the groups with these are the genera. For particular groups, we still don't have a good coverage of knowledge. We also map it in space, etc. Okay, so what this is allowing us is to identify the species that are broadly covered by data. Okay, not only to rely our results on those species for which we have good data for everything, but also to identify which species they are, where are they placed in the phylogeny, and if we've got certain clades or certain genera or certain regions for which we have a deficit in information. And when we interpret results, we limit it to the one we have for which we have good information. Okay, so if something strange appears, in this case, for example, in these two genera, we can say, okay, we don't have enough data. We can call for more data. But instead of assuming that with a handful of species included from these two genera, we have a good coverage of the feeding habits. Okay. All of this together relates to an old idea from Sandbox, which is to create, I don't know if you remember the time of the atlases, but before the advent of Wikipedia and the internet, we have big encyclopedias and big atlases with a lot of maps, thematic maps of the world in our houses. Sandbox proposed back in the 50s to include in those atlases maps of ignorance, maps of we don't know. Okay, this is the first map of ignorance of the history as far as we know. And this is the level of knowledge they had at the time about soils in the world. White is no data. Okay, they suppose no soil, deserts, etc. Now we know there are. I mean, my dryland colleagues get really angry when they see this map. Okay, but then this is just the one. It's very, very, very basic knowledge on the lithology. The two is having some basic idea of the type of soil that is there. The three, it's a classification of soils with some idea of the horizons. And the four is good, it's only here and in Germany. It's a good knowledge of the soils, their horizons, how good they are for different aspects of agriculture, etc. Okay. The main idea is to map not what we know, but to map what we don't know. The idea of Sambot was to put this together with the map of the soils of the world. So we knew that if here in this black area we are saying that there is a spot zone, for example, we know that it's a huge extrapolation that may not be true. This is giving us an idea of the uncertainty of the data. I have to rush on. So also, this also happened in Alagoas. A lot of things happened in Alagoas about this. Richard and myself put together, we first make this call for the need for maps of ignorance, and then Richard and myself got together and look at biodiversity data for biographical purposes. We are guilty of being biogeographers, different perspectives on biogeography, but we are biogeographers. So we're thinking about distributional data, on the distribution of biodiversity. Okay. And we came out with at least 3 factors that compromise the quality of our knowledge of distributions. Survey completeness, how complete and good are our surveys, and the decay of information in time and space. Information that is old is less reliable than information that we gathered yesterday, and the same happens in space. The information that is in the places we have sampled is much better than the information that we are extrapolating to the places we have not sampled. Okay. And we came up together with other colleagues, with Jessica Sarolo, with Juliana, etc., to this main idea: the accuracy of the knowledge about a given taxon, at any level, or community at a given area P is just a function of the quality of the knowledge we have in that area by the completeness. We can have very good quality on an area, but we may lack a complete view of the whole community. But also to the loss of knowledge across time. As time passes, we are losing knowledge, and to the loss of knowledge across space. Okay. Data quality. I mean, Julie is going to talk to you about taxonomic uncertainty, but data quality is quite important. I always use this example. This is a model of the Sasquatch or the Bigfoot. Okay. And this is data retrieved from a database, from 2 databases actually. This is, the points are about shouting, people who heard the Bigfoot, and these footprints are about sightings of footprints. Okay. You make a species distribution model, and you've got the model of the distribution of the Sasquatch. Okay. You make a species distribution model about the black bears there, and they share the same responses to the environment. What happens here? Because, well, either there are some taxonomists that are misidentifying black bear for Sasquatch. These are 2 actual databases online. Okay, this is extreme examples of the people of ufologists and terraplanists and these kind of people. It's an extreme example of all the mistakes we can make as ecologists or biogeographers, not specialized in the group, if we put our misidentifications online. We need to do some quality control on the quality of some control on the quality of those records. Okay. We have been developing several ways of mapping the completeness and coverage of our data. This is how complete were surveys for bryophytes in the Iberian Peninsula across time. Okay, this is the total time, but also across different moments of time. This is what we call the Espanol effect here, because somebody's PhD was around the time that in this region. Okay, but you find to see this interesting pattern when we identify no place or no region of the Iberian Peninsula where we could track the effects of climate change on bryophyte, on mosses. Okay, there was no place. We have good knowledge of some areas, but if we want to track the effect of climate change, we can do that because we don't have this complete inventories in the same place, in the same cells through time. And we also measure climate coverage, how much of the total environmental gradients we are covering with our surveys. This allows us to identify the places that are little covered by data, where our models of biodiversity, species, biodiversity communities will fail, will be uncertain, because they are far from the observation. They are spatially far and climatically far from observation. We have also developed ways of identifying how the information decays with time. Okay, and I'm going to stop on this. This is the paper by Jessica Sarolo. And the things we have been doing lately is to put together all this information, taking information on the different observations and the time when those observations were put together to identify how good is our knowledge in each part of the map, in each grid cell of the map. Okay. In other words, is how informative is our data on the distribution of biodiversity in every place of the map. If we have a lot of information coming from observations that are from other moments of time, from other places, but with similar conditions, good quality, etc., we may have good information on places that we have not visited. Okay, but if we are very far away from the universe of the observation, okay, our models necessarily will be uncertain. And the next step was to create the maps of biographical ignorance. Okay, these are the first example with 3 dung beetle species, and this is the suitability predicted with species treatment models. Okay, and this is the degree of biographical ignorance for each one of these 3 species. The 3 maps look quite similar. They have some differences because they are done with all the data from all the dung beetle species in the Iberian Peninsula. Okay, why? Because dung beetle specialists go to the field and usually capture all species. So the surveys are not directed to just one species or just the other species or to small groups. They are directed to the whole group of species. So we can use information from other species to retrieve this biographical ignorance. Okay. In cases where surveys are directed to particular groups of species, we may have to restrict this. Okay. So we put together this biographical ignorance and predicted suitability, and we can have maps of prediction uncertainty. Okay. In this case, you can see this is the predicted suitability, and this is the level of biographical ignorance. This is kind of counterintuitive. This is we know a lot, low ignorance, we don't know much. And you see here areas where our predictions are that the species is there, and we are quite certain about that. But here you have some purple areas, okay, where our models predict that the species will be present, but there is a high degree of uncertainty about it. Okay, this is what we are trying to do. We are making it virtual. We are identifying when we are interpolating and when we are extrapolating. Okay. The next steps were lined up by Bill and Lennon in 10 years ago, actually, is using these maps of uncertainty that were not available at the time to make a spatial explicit potential error terms. So we can add this to the models in a modeling process, in regression-based models. We can add it as potential error terms. We can include them in Bayesian models as well. We can direct additional surveys. We know the areas where we need more data. Okay. We can do that, or do what we did with these maps, highlight the uncertainty of SDM results and incorporate data uncertainty in the techniques. Okay. It's a bit repetitive, but it's different ways of incorporating uncertainty. So these maps are not only useful to show you that we don't know, we are separating here. We can, the next step is to incorporate them into estimates. Okay. This is another, I'm going to pass this, but we should be adding these to models of global change. And the next step is to create a framework, which is something that we are discussing tomorrow. Create a framework to incorporate these data-driven scenarios in all the process of biodiversity research and management, and find ways of communicating this uncertainty to the stakeholders and the policymakers. This is, I mean, of course, I did not do all of this alone. A lot of to how happy he is. I did this with a lot of people and many more that are not there. So thank you.